Hi, I'm Dr. David Dobson. Welcome to Conversations. Today, my guest is Dr. Vera Sham. Vera is a Cameroonian scholar who recently completed her PhD in plant science from Institute of Molecular Plant Biology, ETH Zurich. Today, we will discuss Vera's PhD research on whole genome duplication in plants. It's a pleasure to have you on my show, Vera.
Thank you very much, David. It's a pleasure to receive your invite and to be here to talk about my PhD journey and project. Thank you.
Vera, what brought your research interest to plant science?
Right. So growing up as a child, I did a lot of farming with my parents and other family members. And I've always had this enthusiasm of how, photosynthetic organisms like plants survive the environmental changes or attacked by biotic and abiotic stresses like heat or drought or insects feeding on them. And so when I wanted to take my bachelor's degree, I had another passion that in future I wanted to contribute to Cameroon economy. And the best way to do that is via agricultural sciences, because that's the backbone of Cameroon economy. And so I ventured into a bachelor's degree in agricultural science. So that's how I started working with plants.
Nice, nice. For your PhD research, you studied polyploid plants. Can you share a little bit about your PhD research project?
Right. There's something really fascinating about, two things fascinating about my PhD research. So it touched on the core of life of organisms. So organisms depend on two things to survive on Earth. One is their genetic makeup, and two is food that is being produced via photosynthesis. Mm-hmm. And so my research that looked at how polyploid plants perform their photosynthesis actually touched those two things. Mm-hmm. So polyploid plants are plants whose genetic makeup have been increased or have been doubled. So they have, for example, they have more gene copies than diploid plants that will have lesser. And when this happens, the entire genetic makeup of the plants is changed. And the plants have to adapt over time to survive the environment. And to survive means you should be able to carry out good photosynthesis to be able to make food while adapting to the climatic changes, especially in this era of global warming that we have. Mm-hmm. And interestingly, those polyploids have been shown to perform very well in very extreme environmental conditions like water limiting conditions. Mm-hmm. So they are very, very important in agriculture. And it's one of the reasons why most breeding and most farmers, they use polyploid plants, but they don't really know why they use polyploids. It's just because these polyploids have these beneficial traits. So some of the traits are they can produce bigger fruit sizes. Mm-hmm. They can produce bigger tubers, like the potato that we eat every day. So the polyploids are bigger than the other diploid plants. And if you plant them, they don't require as much watering, like water supply, as a diploid. Or if there's automatic drought or dry area, those polyploids can survive better than the diploid counterparts. Okay. So those are some of the very interesting characteristics that the polyploid have. But what my research really answered is that everybody knows, oh yeah, the polyploid have this beneficial trait, but they don't understand what changes that occurs inside the plant to be able to give them such beneficial trait, and how that plants have been able to survive, let's say, 30,000 generations, because those genomes do not just double now, they double years ago. Yeah. And they've been able to survive in their environment, and the breeders just go there and they pick those traits, they transfer it to another plant. Nobody has really addressed how come they survive that several generations, what do they have? How do they change their cells, for example? And that's what brought me to the major objective of my research, which was to look at how photosynthetic cells and traits of plants, of those polyploid plants, have changed over time.
Excellent.
And so, yeah, and so I specialize to answer two questions with regards to that. So I first of all look at the cells, the important photosynthetic cells, for example, like stomata, that people don't talk about a lot, but it's very important because it plays a role in photosynthesis. So that's where it when it opens, carbon dioxide can go in and plant can make food. So if it's not functioning, plant cannot make food. The stomata, very important. And it also plays a role in plant transpiration. So it's through the same pore which water is being lost. So it plays a role as well in plant survival in the environment, because if it's open for a longer time, or if it's too big, the plant loses more water and the plant suffers. So my question was to address how the cells have changed in polyploid and how the changes in the cells must have affected photosynthesis as a whole.
Excellent.
Yeah.
So, Vera, how did you conduct your research? I'm sure you spent a lot of time in the lab, no doubt about it.
Yeah, I spent a lot of time because physiological threats, like to measure gases exchange, like the carbon dioxide that I mentioned, that is also very important now on Earth, because if the plant gives so much carbon dioxide on Earth, it's also a problem. And those things are regulated via the stomacher. So to measure things like gas exchange, about the water lost, you need to have technical equipment. And we have some of them. ETH has one that is called the whole plant gas exchange system. So there you can measure gas exchange, and then you can calculate it and measure the approximate photosynthetic rate of that plant. And then while measuring that, you can subject the plants to drought stress. So, and then compare it to the diploid counterpart to see who performs better. So over time, and then I'm working with a new model plant. It's called Arabidopsis arenosa, and trapassen is a common name.
Interesting.
And interestingly, in this plant, we can have diploids. And then in the lab, we can generate this newly synthesized polyploid. And then we can compare them to polyploids that have existed in their natural habitat for thousands of, like 30,000 generations approximately. So when you compare those three things, you can see the immediate effect of how the trait changes when genome doubling recently occurs in those new synthesized plants. And then you can compare how this trait changes over time in those plants that have been surviving in the natural habitat and what they have adapted to. And you'll be intrigued to know that when these polyploids were newly generated, they were really challenged. Yes, they had some advantage, like bigger vigor or hybrid vigor and fruit size, but they could suffer from fertility, their cell size increase. When those cells, for example, when stomata cells becomes bigger, it affects how water is being lost. So more water can be lost, and for those newly synthesized, they cannot really survive water limiting conditions. And you'll be amazed to see how plants standing in the same place, like not moving like humans, but they can change their internal system to be able to adapt to stress environment. So what those polyploids started doing is they started reducing their cell sizes. And then started selecting for particular genes that will help those stomata to function better, because the size have increased, they become slow. So they pick up particular genes over time so that it can help that stomata to function. So they have an advantage of having bigger vigor, bigger size, and then performing better under stress condition. So these are some of the things I looked at.
So what did you learn from your research and experiments?
Yeah. So I learned two major things. So the first thing is that looking at plants with your naked eyes, you will not really see what they can do. But if you take a look at the internal system, using microscopy, for example, I looked at the cell size, I could actually see that plants change their cell size. So their cell size gets bigger. If they want to survive, they would partially reduce that cell. For example, tomato cell, they'll reduce it in size so that they are efficient in whatever they are doing. So that's the first novel, fascinating thing that we put out there. So there's a manuscript coming on that because people have studied this, but they haven't really compared the tree system comparison that they have. And people and researchers have not yet put in place why polyploid have some of the traits that they have. So for example, part of my project focused on looking at why they are drought tolerant. So I discovered that they do two things to be drought tolerant. So they partially reduce the cell size and then they select for particular genes that can help those cells perform better in order to become drought tolerant. And the second thing I learned from the research is those genes that we have identified, can be bred into plants. And that can help in developing plants that are more resilient. Especially in now that we have the global warming, so we can develop resilient plants. And there are genes that we saw that have more than one function, so they can help the plant to be drought tolerant, they can help the plant to improve their fertility, and they can also help the plant to be heat tolerant.
Very interesting.
Very interesting. So when the polyploid, when their whole genetic mechanism changes, they adopt these genes that really becomes beneficial that we can explore in the future to generate this very useful plants.
So I definitely see your research has a huge practical application. So what are the implications of your research?
Okay, for me, I'll start with me.
Okay.
For me and to the general, looking at the African community and background where I coming from in relation to my passion, it has given me deep knowledge in how to advise or tell farmers or explain to them some of the physiological changes they might be seeing in the plant. Like I said in the beginning, the farmer just know, give me that plant. So it doesn't really just know why they are using particular polyploid plant to grow on the farm. And so now I have some basic understanding of that, and so I can explain to them proper that advisory services that this is actually what happens. That's why you have these big fruits, this whatever big cells you're seeing in the plant. So my project has enriched me a lot, and how on that scientific part, and also how to relate my understanding to people in my field and also people that are not in my field. And then the second thing is the implication is what I just talked about, which is using these genes to be bred into plants and to develop plants that are that can better survive this rapid changing environment that we are experiencing right now. And also to be able to understand that it is not all genes in plants that should be mutated, because there's a lot of genetic mutation that is coming up. Even though polyploids have redundant genes, means that they can have 2 gene or 3 gene copy. If you mutate one, the other 2 can function. But there are just some genes that have general function. So if you mutate, like if you delete the particular gene, you can affect fertility of the plant, you can affect how the plant survive to heat, you can affect how the plant survive to drought. So my research has opened that space for people to understand that indeed these genes have multifunction, and so you should be careful if you have to touch them. If you have to transfer them to another plant, then that is something beneficial. Yeah.
Excellent. So many possibilities, and you opened so many
questions. I even encountered this during my PhD defense because when I wrote my thesis, we thought, okay, it's going to be, because I already started with the manuscript, so we thought it's going to be a manuscript kind of thesis, but my advisor was like, okay, let's change this to chapter thesis, because I discovered some new things later. And when my examiners read it, they were like, oh my God, okay, so can we do this in plant physiology? Okay, can we do this?
Wonderful.
And so my advisor was like, stop. Those questions are way out of what she looked at.
It's too much work.
So she was focusing on this one. And that's just because they was fascinated in the questions and gaps that I opened based on what I answered. Because like one examiner could say, I attempted almost the whole of plant physiology because I looked at how plant made food, and I also measure so the amount, for example, the amount of starch that the plant can make based on the differences of this polyploid and diploid plants. Yeah.
So I want to ask you something different. Vera, for your graduate and doctoral studies, you studied in Europe, you left your home. Share with us, what was your experience like?
Okay, that's a good one. So, well, when because I was a very passionate child growing up, so I could say averagely I was a bit more different from my peers as a lady, as an African lady. So some of them wanted a short study to marry and have children, but I've always wanted to study. I didn't think of having a PhD, but I always wanted to be enriched with so much knowledge. And so that is one thing that led me to start applying for scholarships after I had my bachelor's degree. So I started applying for scholarships for my master's. It is not very common that an African young lady, let me not say African, but Cameroonian young lady from the community where I'm coming from, is searching for scholarship by themselves for masters. That can happen, but it can happen either there's an average rich kid that would pay an agency to do that for her, or the parents would just do something and then they can come abroad. But for me, I was an average farmer's child, so I had to sort it out myself. But my parents helped a lot because my mom, in a way, my mom loved my passion, so she would manage a little bit to get my computer, my phone, and everything so that I can do what I know how to do best, searching for these things. And then luckily I got a scholarship at Wageningen University, the best agricultural university in the world. And when I got that, I was so excited.
Wonderful.
Yes, I was happy to come and do my master's there. So I applied like generally, and then study my file, and they're like, oh, maybe this scholarship can help you. And then they gave me the scholarship. That's how I got it, the Anne Wadenband scholarship. I was so excited. My parents were just excited because I was excited, but they didn't understand what the scholarship meant. How would you survive in Europe? You know, even my siblings. But I was just excited I was coming abroad. But as a Cameroonian African woman, there's a question, why is she studying a lot? Why? But how I survived that is I knew where I was going first, and I'm very good at not paying mind to what the society or people around me. If I I don't see it very important. I can exist as if it's not. So some ladies were like, "Oh, Vera, you're so strong. How do you ignore all these voices?"
Yeah.
So I don't
Yeah.
just pay mind to it.
Yeah.
So, and then something happened was that where I got the scholarship, it happened that part of my family lives closer, which is my yeah, they are actually white, they're Dutch and everything, but they have not known me for a while. So when I got the scholarship and I was like, oh, I have the scholarship, and my cousin's mom was like, oh, that's just close to where I live
Okay.
in Netherlands.
Nice.
And that was something very magical that happened to me.
Yeah.
Not every Cameroonian young lady can have such opportunity to have family next by. So when I arrived, despite the fact that they were there, I also faced cultural shock. So the first one I faced was when they asked me at the airport what my postal code from Cameroon. I'm like, what? Postal code? And luckily I had my cousin's mom with me, and she said, yo, she's saying the truth, there's no postal code in Cameroon. That was the first thing that hit me. And then the second thing, and they really helped to serve as a caution, which I appreciate for the rest of my life. They really, my family here, they serve as a caution for me not to really face the intensity of the cultural shock. Because Wageningen as well is a very intensive studious university.
Yeah.
So when I got there, the perception I had from Cameroon was that, oh, when you come to Europe, studies is a bit easier, but studies hit me there like, so the studies was really intense.
Yeah.
I had to dive into it. And then you also have this challenge of you're earning a little bit of scholarship, but you have to manage a little bit to send some money back home for family members and all of those things. That also I really needed to adjust to, because no one back home will understand that you don't earn much. So they just expect, oh, you've gone for scholarship and things like that should be very easy.
Yeah.
And so those were the three things that really shocked me. But then when I finished my master's, I was lucky to, first of all, have like a pre-doctoral studies in the UK. And from the UK, I got this position to do a PhD in ETH Zurich, which you know that is the best in Europe. Yeah. So then I was excited. But at this point, I was a grown-up woman that back home, there's a pressure of, oh, why is she studying so much? What is she going to do with the studies? Is she still studying? And all of those things. Yeah. But then I have this other part of the family as well that really understood where I was going. So I really enjoyed their support, and that helped me to escape the natural African woman pressure that you face while doing this deep scientific studies like me. Because some people in my community did not even just understand what was PhD even. Like, what is PhD? And so now most of them are learning. And interestingly, it has been an eye-opener. So my family is like an eye-opener to the rest of the small community there, that a woman can choose to be or to do whatever she wants to do, so far as it's good and it's not harming people, just let her do it. And then because they can see the outcome of what these studies have brought. And that is what one of my goals is, to use this opportunity to encourage and motivate young Cameroonian women to not set back, to have the courage to go ahead and set up whatever they see that they can do. It's really challenging. Now I can see the challenge. At first, when women say the African culture can be challenging, I thought it was a joke, but because in the middle of a blues, for example, a typical Cameroonian man just hearing how much I have studied, it's automatically just challenge. That woman has studied a lot, and
Mm.
will not even want to hear your opinion because his ego, his everything is just completely challenged. And so you really have to find a way to pass out whatever information you have to do, because Cameroon is still a patriarchal society. So most of the positions or the leading academic positions are all led by men. So we're still struggling to penetrate that part. Yeah.
And Vera, didn't you make a history that you are the first person from Cameroon did a PhD in plant science from ETH Zurich?
Yeah. So that's I didn't know until I had the PhD, and he was like, yeah, if you look at the background, she's the first to earn a PhD in plant sciences from a prestigious university, like ETH. And so it is also part of the story back home, that is also motivating a lot of young girls now. And because some of them really saw how I went to a gradual process. It was not like my family or friends or my parents were really rich, but they saw me grow gradually because of the passion that I had in what I was doing. And so now they're thinking, they can do it. And so that is giving me so much peace of mind. So I'm not even excited because I have this PhD. I'm excited because it has been able to change the concept in the mind of a few people from my community, which is a great thing.
It's such a wonderful story of yours, Vera. It's such an encouragement. Your hard work, your perseverance in breaking all this stereotype and taboos. I really congratulate you on your accomplishment and all the hard work you did. This is really inspiring. Thank you so much.
Thank you, David. This was nice. I've been looking forward to actually express this in a conveyed way. So I'm glad you gave me this opportunity, because it's also a way for me as a medium to continue to motivate my young girls out there, that you can do it, because science and academic is not boring, because growing up here, you're doing science, but it's not boring. So most of the best part of my life I enjoyed by doing sciences, because exposes me to different countries, different cultural backgrounds. I've often had this dream of, if you ask me what my hobby was, I would say, I love traveling, but I've never traveled before. But because of science, I've been in Asia, I've been in Americans, I've been in the UK, I've been in Europe, several countries and stuff. So this is what science have done to me. And it's a complete life service. It's not because there's also a barrier coming back from home, that people think, when you're doing that, you're studying. No, it's a whole, like, it teaches you how to be social. It teaches you how to be humble, because you experience failures as well. And then how to handle those failures and how not to quit. To receive societal criticism, you also have to have the better understanding on how to handle those criticism. And those are all ways of life. So if you have been exposed to all of that, you become a better human in future. And that's what it does to you.
Thank you for sharing. Vera, I want to ask you, what is your advice to those who wish to further research this topic?
Please do, because there's a lot of opportunities with the polyploid plants that is not yet out there. So it's not yet completely understood. Like now, for example, I've looked at these genes that can help plants to perform photosynthesis or to produce their food better, even when they are facing stresses in the environment. What other people can look at is how is to measure whether the survival of this plant does not affect the amount of vigor or the amount of fruit or food that the plants can produce. And people might also want to identify more of those genes that can act as a stack of protection in the plant. So not only about stresses like drought or heat, but you might also look at a gene that can also help the plant to fight diseases or infestant by the insect, because polyploids also have those beneficial traits in them. So those are all the things people can explore.
Yeah, thank you.
Yeah.
You successfully defended your PhD last month.
Yeah.
So once again, congratulations.
Thank you.
What is next for you, Vera?
Okay. I have been keeping this in the basket. Not to talk about it until when I'm there, then I'll unveil the basket. But now David is taking it out of the basket. That's all right. So, interestingly, when I went for one of my conferences during my early days of my PhD, and not early days, but almost towards the end, yeah. And then I did a short presentation, and one of the professors in the field of also working on plant photosynthesis and how plant can protect themselves also against stress, but she looks at excess light. So she saw me, and then she walked up to me and said, "Oh, I would love to have you in my lab as a postdoc whenever you would graduate." And so since she was working in my field, and I thought like that's something I could do after my PhD, when I came back, I had to think about it because she was going to the US and I also had to move. And then I reached out to her. I said, okay, I can take your offer because it's what I am doing. So it's part of plant physiology, so I can work on that. And that's how I got this postdoctoral position.
Congratulations.
Thank you. In Indiana State, in Bloomington. And I plan to start September, October, should everything goes well. But I already have the full position. And the second thing that is interesting in that position is the university. So she introduced me to a body of the university. It's called AfrisNet. Okay. So there they bring in African students on short-term basis to train them on internship or projects and everything. And that nicely fits into my long-term goals to be able to transfer a bit of the knowledge that I have to the African girls. So I'm excited to start there. Yeah.
Once again, thank you so much for your time, Vera, and all the best in your next adventures and endeavors.
Thank you, David. Thanks.